check 2. Hello 2, check 2, 1, 2, check.
Shade it to the mirror, all a Romeo. How am I you? Shade it to the mirror, shade it to the mirror. Lord will lift you high, never circumstance, never ended. There is no like you. Speak indeed, surely comes to pass. All creation trembles at your voice. To the stones I know you will be with me. So I won't be moved. No, I won't be moved. Lord, you are with me. This I know for sure. Send it to the mirror. Send it to the mirror.
Can you go ahead and pray the Holy Ghost?
With due respect, may I humbly request that every one of us here in this hall should please stand for the Vice-Chancellor's procession in respect of the 421st inaugural lecture of this university. Dominic, now please be around here.
Please kindly be seated, please. Thank you very much. You are all welcome to this gathering. The 421st of the inaugural lecture series of Obafemi University, Ile-Ife. This lecture is to be presented by Professor Ezekiel Olugbenga Akinkunmi. And the lecture is titled, The Invincibility of the Invisibles. We are yet to find the ideal magic bullet. Professor Ezekiel Olugbenga Akinkunmi obtained his Bachelor of Pharmacy B.Pharm degree with distinction from Obafemi University in 1997. He subsequently earned both the Master of Science and Doctor of Philosophy degrees in pharmaceutics and pharmaceutical microbiology from the same university in 2004 and 2010, respectively. In further pursuit of professional and clinical excellence, he obtained the Doctor of Pharmacy, PharmD degree from the University of Benin in 2018. Professor Akinkunmi was also a research scholar at the School of Pharmacy, Biosciences and Biotechnology, University of Camerino, where he further strengthened his research expertise and international academic exposure. He commenced his academic career at Obafemi Awolowo University in February 2001 as a junior trainee fellow, and through dedication, scholarly excellence, and distinguished service, rose through the academic ranks to attain the position of professor in October 2018. Professor Akinkunmi is an accomplished scholar, an active researcher whose research interests span infectious diseases, antimicrobial agent development, microbial pathogenicity, antimicrobial resistance, and antimicrobial stewardship. His scholarly contributions have significantly enriched knowledge and practice within the pharmaceutical and biomedical sciences. Within the university system, he has served meritoriously at the departmental level, at faculty and university levels, through active participation in numerous committees and administrative responsibilities. He served as acting head of Department of Pharmaceutics and was also the pioneer acting head of Department of Pharmaceutical Microbiology in the Faculty of Pharmacy. In addition to his institutional contributions, Professor Akinkunmi serves as an external examiner to several universities within and outside Nigeria. He's also a highly respected reviewer for more than 40 reputable academic and professional journals, and serves on the editorial boards of several scholarly publications. Professor Akinkunmi has published over 60 articles in reputable high-impact journals and has contributed extensively to academic books, conference proceedings, professional workshops, and international scientific conferences. He is a fellow of the Pharmaceutical Society of Nigeria and a fellow of the Nigerian Academy of Pharmacy. He is happily married to Mrs. Rachel Olubisi Akinkunmi, and their union is blessed with children. I'm hereby honored to present to you Professor Ezekiel Olugbenga Akinkunmi to deliver his inaugural lecture. Thank you.
Mr. Vice Chancellor, sir, Deputy Vice Chancellors, the Registrar, Bursar, Librarian, colleagues in the academia, non-academic staff, students, other stakeholders in and outside the university system, the general public, gentlemen of the press, ladies and gentlemen. Mr. Vice Chancellor, sir, I stand here today giving glory to the Almighty God who has made this day possible. This is indeed the day the Lord has made. We will rejoice and be glad in it. Today, by the special grace, mercy, love, goodness, kindness, and providence of the Almighty God, I had the singular privilege of presenting this inaugural lecture in the field of pharmaceutical microbiology and biotechnology, the first of its kind from the recently established Department of Pharmaceutical Microbiology, which I also had the privilege of pioneering as the acting head of the department. Prior to the establishment of the department, two great scholars who were my teachers from the Mother Department of Pharmaceutics had earlier presented inaugurals specifically in the field of pharmaceutical microbiology. The first inaugural was presented by my one and only supervisor and mentor, Professor ADEBAYO, who took my hands and led me safely through the rudiments. He presented inaugural lecture on the 15th March 1995 with one of the shortest topics one can have on this platform. The topic was life with. I'm happy that he's here today to see me as a solid testament of his legacy and positive contributions. Following him, on 2nd May 2006, Professor Grace Onawumi presented inaugural lecture series 190 titled Prospects of Plant Products in Antimicrobial Agents. Here is me now, handling the baton with the firm belief that there will be so many others to follow suit in no distant time. I am here to present the 421st inaugural lecture on the title, the invisibility of the invisibles. With the caveat, we are yet to find the ideal magic bullet, serving to provide necessary direction to the inquisitive mind. This is not a statement of failure. Rather, it is a statement of focus and unwavering purpose. The term inaugural lecture comes from the Latin word oratio, which implies that it is a public speech. Therefore, inaugural lectures are described as significant milestones in the careers of professors, serving as a platform for sharing knowledge and fostering connections within the academic and broader community. I need to provide information that I have almost completed working on this lecture long before the pronouncement of my chair in June 2024, backdated to October 2018. Mr. Vice-Chancellor, sir, in this regard, I'm a privileged beneficiary of your exceptional, exemplary, and visionary leadership, for which I'm so grateful. Distinguished audience, please permit me to start with the keyword invisibles. The science of physics and chemistry began in ancient times. Mathematics and biology began even earlier. But the knowledge of tiny living things, the invisibles, their biology and their impact on human lives, known as microbiology, have only been around since the late 19th century. These tiny living things are the ones we now call microorganisms or microbes. They are so called because they are in the size range of micrometers and take space of cubic micrometers, which is far lower than the size which the natural human sense of sight, the naked eye, can easily comprehend. Generally, the human eyes could only see things clearly from the millimeter ranges, which is 1,000 times higher than the micrometer. Bacterial cells are very small, typically in the range of approximately 0.5 to 5 micrometer in size, with diameter usually about 0.5 to 1 micrometer in length for raw cell bacteria of about 1.5 micrometer. For example, the estimated size of an Escherichia coli cell is approximately 1 to 2 micrometer long and 1.5 micrometer wide. For Staphylococcus aureus, it is approximately 1.5 to 1 micrometer in diameter. For comparison, the typical human cell is about 10 to 1,100 micrometer. So bacteria are roughly 10 to 100 times smaller than the human cells. Our eyes need support if we are going to be able to see this organism. That is the role that microscopes have played since the advent of microbiology as a discipline. However, the truth of the matter remains that though you can't see them, these invisibles are everywhere. Viruses, fungi, yeast, protozoan, algae, bacteria, and archaea are truly the proverbial big things that come in small packages. Without these essential organisms, life as we know it wouldn't even exist. As researchers, not only do we know how valuable microorganisms are with regard to human health and disease, but much more for finding solutions to the numerous human problems, such as development of vaccines, cleaning up of oil spills, ensuring healthier food and water, finding treatment for infectious diseases, and development of products to keep our homes clean. Brief historical development of microbiology. Microbiology is study living organisms of microscopic size. The term was first used by the French chemist Louis Pasteur. Robert Hooke, a 17th century English scientist, was the first to use a lens to observe the smallest unit of tissues, which he called cells. Soon after, the Dutch amateur biologist Anthony van Leeuwenhoek otherwise observed what he called amacus with the use of his homemade microscope. He was the first person on record to observe and accurately describe microorganism called amacus in 1676. Because of this extraordinary contribution to microbiology, Anthony van Leeuwenhoek is considered as the father of microbiology. When microorganisms were known to exist, most scientists believed that such simple forms could solely arise from spontaneous generation. The concept of spontaneous generation was finally put to rest by Louis Pasteur in 1858 in an inspired set of experiments in Fpsn, gooseneck flask. The story of Pasteur and microbiology became especially personal because part of his contribution to microbiology arose from his search for the infectious organism, which is typhoid, that caused the death of 3 of his daughters. Mr. Vice-Chancellor, sir, this is evidence that you cannot actually separate a researcher from other aspects of his or her life, most especially the domestic life. The work of Pasteur and the combined effort of many scientists led to the development of the germ theory of disease, for which he became known as the father of modern microbiology. The development of staining technique, such as the differential gram stain by Christian Graham in 1880 and the discovery of electron microscope by Hans Rosca were turning points in the history of microbiology as a discipline, as they brought about the ability to study the structure and functions of the microbial cells. The above are the summarized historical processes that has made the once upon a time invisibles to become visible now, but still yet to the initiated. With the brief history, we can all see that the development of microbiology as we know it today were a result of constant searches by scientists, and that those developments did not come easy. Today, we can isolate the microbes from anywhere because of the pioneering works of these scientists. You can see examples of colonies of bacteria growing on agar plates and fields of gram-stained bacteria observed under the oil immersion lens of the light microscope in our own laboratory. The magic bullet. Once scientists knew that microbes cause disease, it was only a matter of time before medical practices improved dramatically. Surgery used to be dangerous as not doing anything at all. But once aseptic techniques was introduced, recovery rates improved dramatically. Paul Ehrlich in 1904 found the dye trypan red that was active against the trypanosome that causes African sleeping sickness and can be used therapeutically. The dye, with such a dramatic therapeutic effect, providing succor for patients suffering from a disease that was then known to be more or less a death sentence without causing untoward problem to the sick patient, was excitedly referred to as magic bullet by the people of that time. Mr. Vice-Chancellor, sir, this is the origin of the phrase magic bullet in this lecture today. This leads to another important phrase, that is selective toxicity, also on record to be first used by Ehrlich. So the magic bullet is that agent that exhibits selective toxicity to the microbial pathogens. It refers to the particular agent that, while being harmful to the cell and structure of the microbial pathogen, does no harm to the patient. These are the chemical agents we refer to as antimicrobial agents today. Following the success of trypan red in 1910, Ehrlich, in collaboration with Sakharov Ita, a Japanese physician, introduced the drug Fpsn as a treatment for syphilis caused by Treponema pallidum. In 1935, Jan Domark of Germany experimented with numerous synthetic dyes and reported that prone to C, a red dye used for staining leather, was active against pathogenic streptococci and staphylococci in mice, even though it had no effect against that same infectious agent in a test tube. In the same year, two French scientists, Jacques and Teresa, showed the compound Pronto-C was broken down within the body of the animal to sulfanilamide, the true active factor. Domark was awarded Nobel Prize in 1939 for the discovery of the first sulfur drug. The credit of the discovery of first wonder drug penicillin in 1929 goes to Alexander Fleming of England, who discovered the antibiotics completely by accident, a phenomenon which is referred to as serendipity. Sir Fleming, together with the chemists who isolated and defined the penicillin, Fleury and Kane, were given 1945 Nobel Prize in Physiology or Medicine. At the serendipitous discovery of Fleming, many scientists went to town looking for other antibiotics from all available sources. One of those scientists was Simon Waxman at the Rutgers University in USA, who investigated soil from various sources for similar agents that can be used to counter microbial diseases ranging from the world then. Waxman eventually discovered another antibiotic, streptomycin. Antibiotics have been introduced to healthcare. Two of the recently approved first-in-class antibiotics are gepotidacin and lefamulin. Analysis of the antibacterial show that most of these drugs are derivatives of existing antibiotics classes and thus have high tendency to succumb to similar resistance mechanisms. Mr. Vice-Chancellor, sir, distinguished audience, I have classified this antimicrobial agent on the following basis: target organism, mode of action, mechanism of action, scope of activities, clinical uses, and chemical groups. For the purpose of this lecture, I will focus on the classification based on clinical uses and chemical groups. The clinical usage groups are also classified as follows: disinfectants, antiseptics, preservatives, sterilants, and antibiotics. Disinfectants are antimicrobial agents used to control microorganisms on inanimate surfaces. The main target of disinfectant activities are pathogenic organisms, but of course, they kill other organisms too. If you remember those preparations and products that you use to clean your floors, tables, utensils, equipment, restroom, on the hospitals, laboratory, farms, industries, and at home. They are disinfectants. In order to be ideal for use, they are generally expected to have the following properties. They must be potent at their in-use concentration. They must be fast-acting and kill rapidly, and must not be toxic to the human tissue or that of the animals when used in veterinary cases. Also, they must be compatible with other ingredients used in their formulation, must not react with containers or the surfaces on which they be used. They must be stable over a considerable long period of time, especially over the period of their shelf-life and several other properties. Mr. Vice-Chancellor, sir, distinguished audience, we are yet to get the very ideal disinfectants which have all the properties so perfectly. Antiseptics, on the other hand, are agents that are used to control microorganisms on animal surfaces, such as skins and mucous membrane. If you remember those preparations that you use to treat your wound, clean your skin, disinfect your hand, those are antiseptics. They are used on skin, cover every part of the skin surfaces, but especially on hands when they are referred to as hand sanitizers. Preservatives are antimicrobials included in preparation of pharmaceutical, cosmetics, food, and others, such products, to prevent their spoilage and also prevent infections on the host when the particular preparations are used. Sterilants are antimicrobial agents that are used in sterilization procedures. Antibiotics are the golden group of these classifications that most of us are familiar with. They are chemical agents produced by microorganisms or agents derived from these chemical agents, which are used to inhibit or kill other microorganisms and are effective at low concentrations. Antibiotics exploit differences between bacteria and human cells to achieve selective toxicity, targeting processes unique to bacteria such as cell wall synthesis, protein production, nucleic acid replication, metabolic pathways, and membrane integrity. Mr. Vice-Chancellor, sir, we are yet to find the ideal antibiotics against which bacteria cannot develop resistance. Mr. Vice-Chancellor, over the years, and in every country and institution of the world, since the discovery of Pohalic, scientists and researchers have embarked on that search for the ideal magic bullet. In this lecture shortly, I will wish to tell my distinguished audience my contribution to this search for the ideal magic bullet, the successes we have made so far, the reason why the ideal magic bullet is yet to be found, and what needs to be done if we are going to get closer to it. Mr. Vice-Chancellor, sir, I was trained in this university as a pharmacist, the profession that provides solution to the healthcare predicament of men. Pharmacy is the oxygen of healthcare. This is because a health facility without medication in the forms and manner that is useful to the human body is nothing more than a kiosk. Pharmaceutical microbiology as a discipline is the application of microbiology to the field of pharmacy. Simply put, it is that branch of microbiology that studies microorganisms in relation to the manufacture, quality control, and safe use of pharmaceutical products, focusing on preventing contamination, ensuring sterility, and understanding microbial interaction with drugs. By the grace of God, that is my chosen field of expertise for which I'm here to give account of my stewardship. After my undergraduate program, my supervisor, who apparently didn't want me to run away after graduation, gave me some work to do towards my MSc, shortly after commencing my internship in the Department of Pharmacognosy, or the Faculty of Pharmacy. Unfortunately, despite the fact that I completed the MSc within a session of official registration, I had to wait additional years before I could do the other defense of my thesis because of staff industrial actions and under departmental logistics. Following that, my supervisor engaged me in the collection of clinical specimens because there were studies on infectious diseases going on then. I collected sputum and fecal samples simultaneously for close to 2 years. I also collected surgical wound specimens for about 3 years. Unfortunately, the problem of consistent power failure and absence of standard storage facilities in the university at that time made me to lose many of those isolates. Mr. Vice-Chancellor, sir, I lost an estimated number of more than 300 bacterial isolates at various times in the project. It is better not to even discuss the struggles and pain of those times. However, because of the experiences of those times, I'm presently working on making facilities available, powered by modern technologies that will prevent any other student or researcher from having such experiences. Mr. Vice-Chancellor, sir, this will constitute one of the derivatives of our searches over the year. Shortly after that time, my supervisor started discussing the issue of PhD with me. Then the next thing was, what will be the focus of the PhD? Meanwhile, while I was doing identity collections, I was also doing lots of reading around infectious diseases. On a fateful day, I came across a study on Staphylococcus aureus in the stool of infants in one of the top-tier clinical microbiology journals. This looked strange to me because stool is not a normal place where you look for staphylococci. I did literature searches, checking for any study on intestinal staphylococci in any part of Nigeria or even Africa. I could find none. There and then was settled the focus of my PhD. With this, I became one of the pioneers on intestinal staphylococci globally, and the first to report details of this important subject in Nigeria and Africa. Rectal perineal carriages of S. aureus have been documented, that's characterizing the invisibles, have been documented as potential sources of both endogenous and exogenous staphylococcal infections. Akinkunmi and Olugbenga carried out a study in an attempt to provide information about some of the characteristics associated with the carriage of S. aureus in the gastrointestinal tract of Nigerian children. The study suggests that children in Ile-Ife harbor a reservoir of multiple antibiotic-resistant S. aureus strains in their intestine. Through our findings, we recommended that gentamicin should be the first-line drug for treating infections caused by this organism in this age group, and also that the problem demands constant scrutiny. We investigated the carriage of cones in the gastrointestinal tract of children under the age of 3 years in Ile-Ife, Nigeria, and described the isolation of antibiotic-resistant clinically significant species of cones from fecal samples of the children. We concluded that the GIT of children could serve as a reservoir for antibiotic-resistant cones, some of which had reduced susceptibility to vancomycin. We reported a study that identified virulent factors in staphylococci isolated from fecal samples of children and the relationship of the expression of these virulent factors in the S. aureus and cones isolates, production of coagulates, hemolysin, protease, beta-lactamase, and formation of capsule and biofilm, as well as hemagglutination characteristics were investigated. The report showed that cones isolates associated with the gut have similar potential to causing disease as the S. aureus isolate obtained from the GIT tract of children within the study environment. We investigated the pathogenicity of strains of S. epidermidis, which were isolated from the stool samples of apparently healthy children in mice and rats. Results indicated that acute infection in these animals caused temporary behavioral changes, as shown by restlessness and abdominal scratching, but did not result in death, even at a dosage of 2 by 10⁹. Histopathological examination of the affected organs showed congestions, aggregations, and multinucleated hepatocytes in the liver. Infiltration of the kidney tubule intestinal by chronic inflammatory cells coagulates necrosis of the kidney, spleen, intestine, and stomach walls, as well as marked stromal fibrosis of the spleen. This implies that the organism may play essential roles in the function of the GIT. We also noted that there were paucity of information concerning the susceptibility of antiseptics by medicinal-resistant cones, which are of increasing clinical significance. Akinkunmi and Olugbenga reported a study to provide information on this. The micros MR cones were screened against benzalkonium chloride, cetrimide, chlorcidine, gluconase, gentle-valent, and acrylflavin. It was concluded that children physicists can bear importance of MR cones, which reduce susceptibility to some commonly used antiseptic agents, a source that appears to receive little attention. Akinkunmi and Olugbenga reported studies to monitor the susceptibility of S. aureus isolated from fecal samples of apparently healthy children to 3 commonly used antiseptics. The results indicate that there was a direct relationship between resistance to penicillin and a significantly increased susceptibility to antiseptics, even if this decreased susceptibility may not be enough to abrogate the effectiveness of these agents and the concentration of their intended use. The invincibility. The incidences of adverse effects with the use of some antimicrobial agents and the increasing problem associated with the development of antimicrobial resistance, AMR, has shown that we are yet to get the ideal antimicrobial agent that will give the perfect selective toxicity. AMR is a global concern with a significant impact on public health in communities and healthcare settings. Reports indicated that AMR has contributed to approximately 5 million deaths by 2019, and there were predictions that will contribute to approximately 10 million deaths annually by 2050. This equates to approximately 1.4-fold more deaths annually from AML in 2050 than the entire COVID-19 pandemic to date. Mr. Vice Chancellor, sir, distinguished audience, this is the reason for the use of the term invincibility in this lecture today. It refers to the inability of man so far to effectively control these microbes, especially the pathogens that cause problems for us in terms of being responsible for infections and infectious diseases. In spite of all the technological advancements we have presently, infectious diseases still remain one of the main killers of men globally. Vitali Akinkunmi et al. Analyzed the presence of antibiotic-resistant genes and the globally distributed SCC types, subtypes, and fecal isolate of cones in order to examine the genetic drug mechanism, resistant mechanism. Our study was the first report on antibiotic-resistant genes on cones in Nigeria, as well as the first report on the SCC elements in human intestinal cones isolates. Resistant genes were detected only in isolates that are so resistant by phenotypic screening. We concluded that the genetic background discovered in the study could be a reservoir for interspecies gene transfer among cones and isolates in the intestinal tract. Surgical wound infections. Akinkunmi et al. Reported on the microbiological spectrum of postoperative wound infection in Nigerian teaching hospital, and the antimicrobial surgery provides with a view to providing guidelines to the clinicians for making rational decisions over the use of antibiotics in the management of surgical site infections. The infections were polymicrobic and multi-drug resistant. We saw the need for the development of a viable antibiotic policy and drug guidelines to prevent or reduce the indiscriminate use of antibiotics and preserve their effectiveness for better patent management. Akinkunmi and Lamikara studied 28 samples of traditional ophthalmic preparations obtained from various traditional medical practitioners in southwestern Nigeria, western state of Nigeria. They were examined for sterility, clarity, presence of particulate contamination, and pH. None of the samples was found to comply with official specifications, and so we concluded that they were potentially dangerous to the users. The results of poor microbial quality, which we obtained in this study, coupled with other similar reports in some other commercially available non-sterile pharmaceutical preparations in Nigeria, spur us to go further to investigate eye drop offered for sale and used for treatment of eye diseases in Nigeria for their pharmaceutical quality and antimicrobial effectiveness. Eye drop product samples containing gentamicin, chloramphenicol, timolol, tropicamide, tetradosolane, pilocarpine, antasolane, diclofenac, dexamethasone, and flopyprofene were included. The study concluded that generally, multi-dose eye drop products offered for sale in Nigeria were of acceptable microbial quality and possess good antimicrobial effectiveness. As expected, the results from this study, of course, gave us much relief. Mr. Vice Chancellor, sir, my collaborators in the Department of Chemistry, we have worked together investigating, establishing, and confirming the antimicrobial properties of so many agents, natural, synthetic, old, and novel. For about 2 decades now, my laboratory has been like that of Nobel laureate Jan Dosma and Selma Waxman, performing various degrees of scientific scrutiny for many well-characterized chemical agents while characterizing the organism against which they have been tested. I'm happy to report, Mr. Vice Chancellor, sir, and my highly distinguished audience, that the collaborations are still ongoing and we are making good accounts of them, finding candidates with good promises for development as chemotherapeutic agents. Hopefully, one day, with adequate support and funding, this university will get our own penicillin or cetomycin. One of the groups of synthetic agents that we are scrutinizing for potential as magic bullets are the coordination compounds of amino acid. Finding literature reports indicating their potential in this direction. Ayila Akinkunmi et al. Synthesized, characterized, and screened several compounds of glycine, phenylalanine, and aspartic acid in various studies against reference and clinical strains of drug-sensitive and multi-drug resistant strains of bacteria, such as MRSA and carbapenem-resistant strains. Results indicated that the complexes showed considerably higher activities than the free ligands. Some of the complexes demonstrated better activities against one or more of the tested microbes than the standard antimicrobial agents used as controls. In a recent paper, Akinkunmi et al. Reported the antibacterial spectrum of some aspartic acid complexes, indicating that the complexes have broad spectrum of activities against both susceptible and cyclically resistant strains of bacteria, including those known to be opportunistic pathogens. Clearly, we are bringing the complexes closer and closer to the clinic. Mr. Vice-Chancellor, sir, distinguished audience, it's a matter of time. Fuller details of these interesting stories should be given in future inaugural lectures by my collaborators. So, I hand the story here for now. Plants have always been contributed largely to medicines and healthcare preparations by providing lead compounds for drug development or as refined herbal remedies. In our continued efforts for potent antioxidant and antimicrobial agents from natural sources, we have investigated from some selected plants, their extract and their bioactive constituents. Furthermore, some of the plants were formulated into dosage forms in order to move them closer to clinical applications. We investigated the extract obtained from anacardium accidentally leaf for antioxidant and antimicrobial activities. Bioacid-guided fractionation resulted in the isolation of agatis flavone as well as a mixture of quercetin 3-rutinoside and quercetin 3-rhamnoside. The isolated compound possess essentially inhibitory rather than cider activities against this organism, indicating the type of use they can be put into on further development. Matularia acuminata is a small tree or shrub or tropical rainforest. The leaves are used in Nigerian ethnomedicine for the treatment of microbial infection. Olugbenga et al. Reported studies that determine the most antimicrobial activity fraction of the leaf and the constituent responsible for the activities. The isolation of a new bioactive phenolic glycoside and a saphonin nearly described as matularin that demonstrated antioxidant and antimicrobial activities may be responsible for the activities of leaf extract and its ethyl acetate fraction were reported. Ajayi Adirogba Akinkunmi et al. Reported the isolation of antioxidant compounds from the ethyl acetate fraction or the leaf extract of nuclear latifolia. The compounds are trans-34 dihydrocinamic acid, quercetin, quercetin 3-glycopyronoside, and chlorogenic acid. One of the significant results of note from the study was the activities of 3,5-O chlorogenic acid, which had an MIC of 1.25 milligram per ml against Pseudomonas aeruginosa. These agents could therefore be effective in the management of oxidative stress-related infectious diseases. We have also reported antimicrobial activities of metabolites, extracts, and formulation from Richia caparoides, alternata dentata, Denetia tripetala, Chrysophyllum albidum, Xanthoxylon xanthoxylodis, Citrus sinensis, as well as antiseptic herbal soup formulations of combined leaf extract of Senna lata and Eugenia uniflora. Essential oil of pharmaceutical formulations. Essential oils constitute major extracts from plants used as antimicrobial agents, and the biological effect of essential oil have been attributed to the of phytochemicals. We did a detailed investigation on the composition and antimicrobial activities of freshly extracted essential oil of 6 citratus and oils stored up for up to 9 years. The MIC and MBC of the 3 stored oil were similar. Antifungal activities were higher in the stored oil. We also found that the storage of the oil made available many components that were not found in the fresh oil. Hence, we concluded that the antimicrobial activity observed for the oil samples were due to different constituents. Oladimeji et al. Reported the development of an effective topical formulation of the oil of Leafia multiflora, Modenk, for the treatment of skin infection. Results showed that the Leafia ointment formulation showed greater antimicrobial activity than salicylic acid ointments used as controls, suggesting the potential of these formulations for further development. Mr. Vice-Chancellor, sir, distinguished audience, please permit me to reiterate here again that as a matter of time, fuller details of these interesting stories will be the component of yet further inaugural lectures. So again, let me hang this story here for now. We are presently working on a grand challenge in Nigeria sponsored project, developing locally produced O-creatinum oil-based cell called Osimum oil gels as alternative to chlorhexidine gel for the prevention and treatment of varicosities in Nigeria. The GCNG indicated that it is empowering the brightest minds in the country to solve Nigeria's biggest problem challenges in these sectors. Mr. Vice-Chancellor, sir, my distinguished audience, this singular project represented most appropriately a value recognition and confirmation of our position as one of the global innovation leaders in this ecosystem. In a recent visit to the university, the CGCNG indicated their interest in building a center of excellence in the university because they were highly impressed by the progress of our work. We expressed our gratitude to the National Society of Nigeria and the GCNG. In a recent narrative review, Miller summarized the therapeutic approaches under development to tackle EMR. These strategies have been devised at local, national, international scales under a multidisciplinary One Health approach. This includes surveillance, antimicrobial stewardship, infection prevention resources, therapeutic innovation. The approaches also include the use of vaccines, pathogen therapy, immunotherapies, microbiome-based therapies, antimicrobial tonodynamic therapies, nucleic oxide-based treatments, nanomaterial approaches, and enabling technologies such as artificial intelligence and organ-on-a-chip systems. Many of these options remain early stage, and with hindsight of history, we can be sure that bacteria may eventually evolve around them. But these innovations reflect a powerful shift. Instead of betting on discovering a single antibiotic or a magic bullet to address resistance, researchers are building a more diverse and resilient toolkit to fight antibiotic-resistant pathogenic bacteria. Suggestions and recommendations. As I close this presentation, distinguished audience, ladies and gentlemen, I wish to give additional reasons why the ID magic bullets are essentially effective, and by that, make suggestions on possible ways of addressing it. Research and development challenges. R&D in antibiotics is risky business because new antibiotics are used sparingly in order to preserve their effectiveness. Companies often sell too few doses to recoup development costs, even after the regulatory agencies approve the drugs. Technology challenge. With emerging technologies, there's need for continuous capacity building and upgrade of research and innovative facilities to do cutting-edge studies. As I talk of technology challenge, I remember an incident that happened while I was conducting my PhD project. The phenotypic approach I was using for certain aspects of the work was saying something strange and interesting. Unfortunately, I didn't have the necessary facility to conduct molecular confirmatory tests for my observations. A few weeks later, I found a report of same result as a very significant novel discovery with the molecular mechanism well documented by another research group abroad. Well, it is exciting to know that the kind of facilities that I needed then are now available closer home today. Innovation support challenge. These kinds of studies need adequate support, institutional, infrastructural, and finance. Without this, nothing gets done. Situations where researchers use their salaries to conduct researches cannot bring the needed breakthroughs the nation needs. We thank our university for the encouraging institutional support available now, especially through the Central Office of Research. We wish we had such when we started our research career. Now, sustainability is the key. Personal challenge. There are no people to do these works. There have been serious challenges of both number and quality in this respect. For example, in the Department of Pharmaceutical Microbiology, we are seriously short of able hands to combine research with teaching to make the necessary impact in this field. Something has to be done urgently to get capable young blood who will take this kind of work further, especially those who are trained in the art of the apothecary, the pharmacist, who can feel the nature of the eventual medicinal end product from the very beginning of the research and innovation by virtue of training. Presently, it has become difficult for staff to take research leave or career development and capacity building because if they go, who will take the lectures? There's a need to do everything possible to encourage versatile graduating students to come back to build the system. This was what made me to eventually decide to stay back, despite my having tremendous opportunity to JAPA immediately after my first degree. In love and concern for my welfare and progress, Being aware of the innate potential they knew I had, my family made all possible facilities available for me to join them abroad. Following up with me for years, they had to leave me alone when I was not forthcoming. The only thing I can do today is to apologize for disappointments. It was because then, although it was not really very clear to me, and I didn't know how to express myself then, I sensed a higher calling to stay back, give back, and build a system that raised me. Because I saw even then, there are no people to do these works. Mr. Vice-Chancellor, sir, distinguished audience, at this juncture, let me present my modest contributions in other areas of the duties of academics, which has to do with teaching and community services. I joined the Obafemi University in the year 2001 and rose through the rank to professor in October 2018. Over the years, which have been full of up and down, I've had the privilege of supervising over 200 undergraduate students. I've supervised some MSc, MPhil, and PhD students, with each of them presently doing well in their various fields of practice. I'm a member of several prestigious academic and professional bodies. In the advent of COVID-19, I conducted the very first documented online teaching in pharmacy students, shortly after school were closed down by government. Our experiences was published in the Pharmacy Education Journal of the FIP in April 2020, representing the first report of such experience for Nigeria in the global pharmacy education landscape. In order to support the effective teaching of pharmaceutical microbiology courses, I published a comprehensive review of the third edition of the textbook, Essential Microbiology by ADEBAYO. A fourth edition of the same book is now available, published in 2025. I got a Keystone Symposia Global S-Series Scholarship to attend the conference in New Delhi, India in 2011. I was a research scholar at the School of Pharmacy, Bioscience, and Biotechnology, in fact of Camerino, Italy, in the year 2011. I have received certificate of merit from the Federal Road Safety Corps, Merit Award on the PHN of Sun State, Leadership Award by the National Body of Pharmaceutical Association of Nigerian Students, PANS, Excellent Service Award by the Association of Hospital and Administrative Pharmacy of Nigeria. I have received certificate of commendation by the Association of Industrial Pharmacists of Nigeria. I have received award of excellence by the Association of Community Pharmacy in Nigeria. I am a recipient of some research grant, either as a co-investigator or main researcher. Among these are the TEFON Israel-based research grants and the ongoing GCNG grant. I have served as either chairman or member of several committees at the department, faculty, and university levels. I serve as the acting head of the Department of Pharmaceutics and pioneer acting head, Department of Pharmaceutical Microbiology. I have also served the dean on the board of College of Health Sciences and several faculties in the university. In the university, I have served as associate staff and resource person for the Central Science Laboratory since year 2010. I was a consultant for the OAU Investment Company Limited for several years on pro bono basis. I have served and I am still serving as a university examiner for master's and PhD thesis in several departments in university. I also serve as external examiner to university within and outside Nigeria. I was a one-time chairman, electoral committee of OAU Star School Parent Teacher Association, executive, and also serve as member of the Star School PTA, representative on crisis resolution in the school. Presently, I am the chairman of the PTA of Obafemi Awolowo School, serving with my ever supportive and visionary executive and representing my co-parent as a member of the governing council of the school. It has been a wonderful experience. My contribution to the pharmacy profession varied from education to practice to regulation to leadership. I played active roles at all levels of pharmaceutical society of Nigeria, from the state to the national level. I salute all men of honor, colleagues, pharmacists in the house. Thank you for joining me to celebrate this occasion. I salute all the leaders in the profession who are witnessing this occasion, either by their highly cherished and appreciated physical presence or virtually. I hereby recognize the chairman of Psn, Pharmacist Michael Omoneyi, the president of Nigeria Academy of Pharmacy, ably represented Professor Larry Bale, and the president of the PSN, Pharmacist Tanko Ayuba, the president with whom I served during my time as the national chairman of NAPA is here, Professor Siri Usifu. Mr. Vice Chancellor, sir, I'm a fellow of the Pharmaceutical Society of Nigeria, and I'm a fellow of the Nigeria Academy of Pharmacy, and the first to stand on this podium in this prestigious institution after having been conferred with those two combined prestigious professional recognition. All glory be to God. As an academic, my technical group is the Nigeria Society of Pharmacy in Academia. During my service as National Chairman of NAPA, among other things, we collaborated with Pharmacy Council of Nigeria to establish the first time standard for academic pharmacy, which is now published in the book, Minimum Standard for Pharmacy Practice, published by the PCN. We also established NAPA Outstanding Pharmacy Student Award, which is being presented in all pharmacy schools in Nigeria since 2023. A word of appreciation to all members of the executive and branch leaders who served together with me in those challenging years. Those are the years I was crisscrossing the country, serving my association. My contribution extended to the community. In various ways, I am a community man. And I thank God for every community where I've lived and in the course of my career. Mr. Vice Chancellor, distinguished audience, I am a singer, composer of songs, and author of music. Some of my songs are presently available on many online music outlets, and this on YouTube. Indeed, I can say what makes so many people familiar with me are my songs, which used to fill the airwaves of NTA many years ago. Brief reflections. Mr. Vice Chancellor, sir, I have stories to tell, because somebody like me cannot be here today without stories. I have earlier alluded to the loose pastor, the father of modern microbiology, whose tremendous and captivating historical contribution to the field of microbiology and healthcare became interesting because of the many dimensions of his life. Indeed, without these dimensions, the world might not have had the loose pastor and the blessing that he has known it. They say opportunity comes but once. Using this good opportunity within the short time I have and the presence of this distinguished audience, I will make an attempt to share some parts of the story behind the glory, but I will be brief. Let me start by giving glory to the only invisible and invisible one, the one who, even though we do not see, the evidences of his presence are so glaring in people like me. In my rumination, I was wondering what the size of God is. We cannot call him a micro because no microscope or any other technology of man can detect him. His invisibility is not because he is small. It is because he is big. If I can referently borrow that word to use, he is bigger than the eyes can see and brain can comprehend. We cannot see him because we are in him. In him we live, in him we move, and in him we have our being. For how can our invisible E. coli, hiding somewhere in the infinitesimal part of the intestine of man, or an invisible epidermal cell, hiding somewhere on the human skin, claim to see the man inside whom he stays? Comparatively, we are far smaller than the E. coli or staphylococcal cell in the planetary earth, which is less than a pin drop in the universe. Trying to envisage about Einstein in my theoretical mind, Mr. Vice Chancellor, sir, compared with the only invisible and visible one, I can see the whole of our earthly planetary system as equivalent of the smallest organism in the microbial cell, and all that we do on this planet are just like the metabolite processes in that organism. Again, just as the human DNA, which is tightly packed supercoiled in the invisible cell, has a length of approximately 70 billion kilometers when fully extended, long enough to travel back and forth between the Earth and the Sun hundreds of times. As the invisible God, who filled the immeasurable universe, true and true, became the flesh we can see in Christ Jesus. Mr. Vice Chancellor, sir, I am not saying all this because I'm a scientist alone, but just like some of the scientists that have come before me, such as Gregor Mendel, the father of genetics in biology, and Isaac Newton, who with the famous Newton law of motion is also regarded as the father of mechanics in physics. I am a cleric with formal training in theology, coupled with substantial material experiences. Sir, God is too much. He is simply indescribable. Mr. Vice Chancellor, sir, I am a proud native of Oyo Town, where I have both my primary and secondary school education. My primary education was in early primary school, Kelola, Oyo. This was school where I cut my teeth as a puppy. My full appreciation got to Oyo State government at the time, who gave me free education in the full complement of the book and stationeries we needed in primary school then. I remember vividly, today my primary one teacher, Mrs. Afolabi. May God bless her for me wherever she is. I also remember the headmistress then, who used to carry me in her car whenever she met me on the road, walking to school. That singular privilege made me to enjoy the privilege of undue respect and protection from fellow pupils who were possibly bullying me then. For who did his parents born well to bully the son of the headmistress? My alighting from her car in the presence of everyone made them to think I was her son. Unfortunately, by the time I got to primary 2, I did not see my mother again. It was later I learned that there was something they call transfer of teachers. May God bless my mother and her offspring wherever she is presently. I also remember Mr. Titebi, my primary 2 teacher, and Mr. Rufai, one of my primary 2 teachers. Now I remember vividly one day during a class in primary 4 that our new class teacher who told me that I was going to primary 5 that day. To cut the long story short, that was how I joined them in primary 5 in the 3rd time to be promoted to primary 6 without any input from my parents. The teacher the Lord used to perform that miracle is here today. Papa Akinkunmi Opabomi. I also thank his friend, Mr. Debomi, primary 5. Following the primary school, I proceeded to the secondary school with the best school anthem in the world, Ile-Ife High School in Oyo. I thank Mr. Makanjola, my class teacher, and I also thank one of the teachers I represented here today. My biology teacher in secondary school is here, Baba Paul Osuntola, representing my secondary school along with others in many competitions. I'm bringing trophies home. I have high respect for teachers. That's why I bring them. And I don't bring chief and kings. Recognize the presence of members of the old and student association of my acknowledgement. I celebrate my lecturers, right from the Faculty of Science to the Faculty of Pharmacy. Professor ADEBAYO, my one and only supervisor, deserves a special mention for his tutelage. Professor Lamekara supervised my BPharm, my MSc, and PhD degrees. I share this unique patrimony with Professor Iruka Okeke, presently of the Faculty of Pharmacy, University of Ibadan. I thank all my teachers, all members of the Department of Pharmaceutics, Professor Nawumi, Professor Ravitiya, Alebiowo, Oladimeji Oyedele, Dr. Adetogun, Osungona, Akinkunmi, and Olasayinde. I remember my colleagues and office mates of blessed memory, Dr. Mrs. Oluwatoyin Abibola Igbenegu. May the Lord bless her wherever she is. I thank all the deans from my student days, Emeritus Professor Festo Ogunbona, Elojoba, Ogundaini, Olugbade, Bolaji, Adebayo, Aaron. Incidentally, Professor Aaron did his valedictory today. He handed over both baton to me. Today is very significant. I see like somebody going on and said, Akinkunmi, the faculty is in your hand. And I recognize my present name, Professor J.O. Soyinka. I celebrate all my colleagues in the faculty and outside of it. All friends and classmates, 1997 class, some of whom are here today. It's like yesterday when we were attending lectures together in Said, Marcus Lecture Theater. All glory be to God. I thank all my colleagues in the Department of Chemistry, Professor Aderoba, Ajayi, Olabanji, Adenoga, Ojo, Ayelabola, Ajile, Ajayi, all of you, I recognize you. I celebrate all my pastors, represented by the President of Nigeria Baptist Convention, Reverend Dr. Israel Adelania Kanji, and I thank all my, represented by my Lord Bishop there, and my brethren, churches, where I have had the privilege of worship and ministry. Taking my physical condition, I can confidently say that I am more or less a family project. As you see me in front here, I'm representing a group of people who paid the sacrifice for me to be here. I'm representing the great Akinkunmi family from my late and beloved grandfather, my grandmother, Adola Ajike Akinkunmi, who raised me and my sister, Omalola Moses, who is here, and my dear cousin and brother, Olusoji Oluwalana. I thank my daddy, Dr. James Oluwale Akinkunmi, and his family in the US, without whom my education dream would possibly have been aborted. I thank Uncle Philip Ayo Akinkunmi, who was my chief examiner at home whenever I came back home from Lagos. during my secondary school days. I thank Uncle Rotimi, I thank late Mama Dwekeside, Mama Mariana Akinkunmi, and all my brothers and sisters. And I thank my in-laws, the Olayiwolas, Olorun Semikpes, Olorun Seguns, and Okunades. These are the people that helped me to carry logo when I could not carry, so I have to appreciate them. I appreciate Mr. Esther Olaniyan, Olaniyan, Mayowa Adeoni, David Oyewo, Jumake Olorunleke, and BSF OAU for their contributions to the success of this lecture. I wish to thank specifically Pharmacist Adeolu Adediran for his support over the years. Mr. Vice-Chancellor, sir, I'm privileged by the grace of God to have both my father and mother here today. Mr. Samuel Onalapo Akinkunmi is here. Mrs. Victoria Akinkunmi are both here. I am not one of those people whose father and mother are late. They are all here alive. All glory be to God. I thank God for the various, that is a sign that I will live long. I thank for the various roles they played in my life. I'm happy for them too. You will live long to see the goodness of the Lord in the land of the living. Finally, I thank my children and my wife. Testimony Orin Ayo Akinkunmi and I did all my heartily research work together, including PhD. I remember in those days, it would slip off at the laboratory while I work in the lab. It was later joined by my fellow Yebi Akinkunmi. I thank Blessed Orin Ayo Akinkunmi, my gang, glorious Awolowo Akinkunmi, who have been such a great delight, contributing tremendously to what I am today, in their own special way and unique ways. Children, you know Daddy love you, irrespective. Finally, I will greet my wife, who despite seeing me clearly, married me totally, fully, completely, physically, spiritually, socially, emotionally, financially, domestically, corporately, reproductively, unapologetically, unequivocally. My dear wife, Rachel Olubisa Akinkunmi, I love you agapely. Mr. Vice-Chancellor, sir, my dear audience, even though through this lecture today, I have presented a reality that we are yet to find the ideal magic bullet, I can tell you with all the confidence in me that I have found an ideal wife. Thank you very much for listening.
Okay, thank you. Obafemi University Ile-Ife, Certificate of Presentation of Inaugural Lecture. This certifies that Ezekiel Olugbenga Akinkunmi, a professor of pharmaceutical microbiology and biotechnology in the Department of Pharmaceutical Microbiology, Faculty of Pharmacy, Obafemi University Ile-Ife, delivered the 421st inaugural lecture titled The Invisibility of the Invisibles. We are yet to find the ideal magic bullet. On this day, May 26, 2026. Congratulations.
Yeah. Just roll from side to side like this. The sure mercies of David. Tàbí torí mo mọ àdúrà àgbà tàbí torí gbogbo ìwé tí mo kà rè àánú Olúwa tó gbé mi ró mo ní kí n máa ṣe tí mo kà sẹ́.